My dad thought you know how it's like
I reviewed unit 1 yesterday, and I think one that I'm gonna really need to one that I'm gonna really need to look at is and the difference between eukaryotes and eukaryotes, because I know for a fact eukaryotes have a nucleus, but eukaryotes don't, and I'm having trouble connecting them with like bacilli and cocci, because I think bacilli can only be gram-positive.
Well, it depends. So that's not the shape of it. So yes and no. So bacilli, when we're talking about the shape of a cell, that just refers to the same shape of the cell.
Yeah.
But if we're talking about organisms that are named bacilli, they have that in their name. They are endospore forming.
Yeah.
All endospore forming are gram-positive.
So if it's in like let's say for example, bacilli. I know it's probably not a correct name, but.
So yeah, if your name was Bacillus anthracis, then that's a gram-positive for sure. Yes.
That means it'd be an endospore, and it would have a nucleus.
Yeah. No.
Oh wait, no.
No nucleus.
No nucleus.
None of the bacteria have nuclei. None of them.
All of them are eukaryotic.
Yes.
Because they don't have nuclei, but they have things like proteins, no?
They have protein, they have ribosomes, they have nucleic acids, they just don't have membrane-bound organelles.
So they don't have nuclei, mitochondria, and the other one that I always forget the name of.
The endoplasmic reticulum.
Yeah.
oh, it's so great.
All right, everybody get signed in on the Kahoot so we can get reviewing chapter 20. Our lecture day is over chapter 21, documenting the diseases of the cardiovascular and lymphatic system. Today will be our last lab, like our last wet lab. So lab 18, and then next Monday we'll do the lab review in the lab, and then next Wednesday is your lab final. So
Because we're not inoculating today.
So there's only 1 week left in the semester after that. That last week, that's such a semester, I sent you guys an announcement about that one. Week 16 of the semester, we don't have lab. But I will be having, on Monday of week 16, so the week right before finals week, Monday of that week, we will be reviewing the content that was the most difficult for people. We'll be going over genetics, we'll be going over aerobic respiration and metabolism, and the immune system. So I'll be giving a review for that in the lab time on Monday. On Wednesday, we will be going over some of the meta questions that we'll start working into, like more talking about, okay, if I ask you if you step on the nail and the bacteria get in, like our question that I provided you guys, like talking through how to start applying that stuff. And I'm going to expect you guys to engage in that stuff if you're coming on that last week. It is optional attendance. I will not be recording it. And if you don't go, then it's on you to learn your concepts. Remember that in this class, I expect you to learn the content, not just study and memorize it. There's a difference there. And even if I were to ask you guys, and that's what, like when we go back to the note-taking module on my Canvas page, and you could go through that and use that for note-taking. And I mean, I would have highly recommended that you'd done that at the beginning of the semester. And if you are now just looking back on that, then that means a lot of us have some work to do to go back to units 1 and 2 and learn that stuff and not just rely on recall, because guaranteed that's not going to get you through my final. Yes.
And on the calendar section, it says the final exam is like posted on Tuesday. Is it Monday?
It's due on Tuesday. So I have it like for all of my sections. Okay. It'll be due on Tuesday. So all of my sections will be done with the final exam on Tuesday. You guys will have yours on Monday in class during the normal class period on finals week. So finals week, Monday, come in here, same time, same place, 10 a.m., just like our other exams. It'll be a 100 questions Yes, it's previously been 50, but you will have 100 questions on your final exam. And we should have the time blocked out where we won't have, if you ended up going over time, like I'm pretty much gonna let you finish it. We should have plenty of time for you guys to finish it. There's no lab afterwards, you know, so we will have all the time to get that done. There will be questions on the exam that will be similar to like what we've been talking about in class, where if I say, okay, your organism comes in, your patient comes in and they have the signs and symptoms of meningitis, you look at their CSF and you see in there that the pathogen has a nucleus and mitochondria and organelles that are membrane bound. If you see that, which of the following choices could be the cause? And if I give you choices, some of them bacteria, some of them fungi, and some of them viruses, you should know that if you saw the pathogen had organelles, that it has to be the fungi choices.
Yeah. Bacteria, viruses,
Yeah.
they don't like eukaryotes.
Yep, that's a unit 1 concept. So that's the type of understanding I'm expecting on the final, and you need to expect a good portion of those questions to be like that. I don't mean like 3. I mean like probably half of the questions on the exam will be like that. All right? This is stuff that if you look back in my notes, and you're looking at my notes and you're looking for word recall, and you're used to saying, oh, this was the definition, this was the exact word for word definition, pick up your big girl panties and put them on and learn how to learn. Understand the stuff. Don't just regurgitate back to me the word. If you can explain it to me. So if I were to say to you guys, okay, well, we saw that the cells had organelles and stuff. And so I would choose maybe, Coccidioides immitis, which is one of our fungal causes of meningitis. Maybe that was the choice. And so I would have chosen that. And then you would mark down your little paper, okay, meningitis, if it has organelles, Coccidioides immitis. Why would I choose that compared to the other one? Can you explain to me why the other ones weren't the choice? That sort of stuff. So you need to be analyzing every question from every test or every homework or Kahoot or Quizlet that you're using. Tell me why it's the right answer. Why is it the wrong answer? That sort of stuff. You should be able to explain it to me. Yes. So the number 1 advice that I gave you guys at the beginning of the semester was during my lectures, you get my lecture slides and you guys are writing notes on my slide. Like I write down a little jot of something. Okay, so this slide was talking about this and I'm writing down these things, right? After my lecture, go back and take your notes, however you took them, your notes on my slides, or if you're writing all handwritten notes or whatever it is, take those notes and now reorganize them into a study sheet. In your own words, do not just recopy it down the words. In your own words, summarize the content in your study sheet. Guaranteed, that's going to get you farther than any flashcard ever will. And none of you, I bet most of you aren't doing that. So there still might be work to determine what the best way to do that is. If you're copying and pasting stuff, if you're having ChatGPT write that stuff for you, no. In your own words, explain it. With your brain, think through it and explain it. I mean, you could try to trust ChatGPT. ChatGPT might give you correct information, but it thinking through that and getting you the answer is not you thinking through that and getting you the answer. And if you can't explain it verbally to me, you cannot understand the concept well enough, period. So when I ask you guys, okay, if we have a flu infection, and we start thinking about flu getting into your body, we start talking about immunology, which is something that everybody's the worst at, right? So we start talking about immunology with flu getting into your body. All right, so we might be thinking initially flu inside, that's going to be CD8 response. Yeah, you're right. It will be ultimately. But how do you have to activate the CD8 response? You need to have the CD4 cells. And how those CD4 cells activated?
By the T helper cells activating the B cells.
Yes.
That's making antibodies, right?
So B cells do make antibodies, but how are the CD8 cells activated? What cells activate the CD8 cells? TH1. All right, what are TH1 cells? We're getting there. What are TH1 cells? They're T helper cells. I heard somebody say that. They're the captains. Right, so tell me about them.
Yeah.
What's important about them?
From the captains to activate those tankers
Right.
and bombs.
So sure, yes. Do they have any certain molecules on their surfaces that help them do their job or get activated or anything like that? No, no, they don't have those on their surfaces. This is what I'm talking about. You see what I mean? You don't understand it. That is a sign you don't understand it. So that's not me being mean, that's just saying the fact. That is stuff that you guys need to go back to. That's what I expect you guys to understand it and explain it back to me. Not recall or regurgitate my words. That's proof. So T helper cells, they have CD4 on them. Nobody said that. At least I didn't hear it if you did. They have CD4 on them. T helper cells do. CD4 is a molecule that recognizes MHC2 presenting antigen. CD4 recognizes MHC2. MHC2 is not on the T helper cell. It's on the antigen presenting cell. Only antigen presenting cells can have MHC2. Anybody know what our antigen presenting cells are? Nope.
Nope.
Antigen presenting cells, the dendrites, the macrophages.
B cells can do it too.
Yes.
So those are our antigen presenting cells.
Aren't those that?
They will take outside and put it on MHC2. CD4 recognizes that it is MHC2. That's what CD4 does. And so the T cell receptor, what does that do?
The T cell receptor.
Yes. So when we have an antigen presenting cell, let's say it's a macrophage. You can agree that is one, right? It picked up something from outside.
Yep.
It picks up a thing, engulfs it for phagocytosis, puts the outside thing on MHC2.
Puts it up for oxygen.
Yes.
Yeah.
All right. So
All cells have MHC1.
What
Every
cell-
radical
right.
one.
yes. So now we're just saying things though, but still stay on what we're talking about with this particular scenario. So we picked up an outside thing as a macrophage. We put it on MHC2 because it's from outside. And I'm holding it up. One thing I was explaining to one of my students last night, MHC stands for major histocompatibility complex, but you can also think of it MHC in your own mind. Think of it as the manhandling complex. It's the manhandling guy. It's going to manhandle any antigen. If it came from outside, MHC2 manhandles it. If it came from inside, MHC1 manhandles it. Any antigen, not specific, any antigen. If it came from inside or outside, right? So this one came from outside. MHC2 is manhandling it. Now what?
So now it's up for oxygen. Does anybody recognize this?
Mhm. What's going to recognize it?
T cell receptor.
The T cell receptor. So what's the T cell receptor interacting with specifically? The antigen. And it is a specific interaction. So when the T cell receptor sees that antigen, it sees that antigen, not any antigen, specifically this one. And then when that happens, CD4 recognizes this is on MHC2. It's from outside. This is the correct response of what's supposed to be happening. Then that T helper cell gets activated. Are we starting to think through this a little bit better? Okay. So that's the type of thinking I want you to apply to all the concepts that we've been learning. But it's a lot, I understand. But if you sucked at immunology specifically, focus on the stuff, and it's crunch time, y'all. If you think that like the week before the exam is crunch time, I hate to break it to you, it's crunch time now, especially if you were relying on the study guide. So it's crunch time. So you want to go through unit 1, 2, and 3. We're doing unit 4 together, and unit 4 is a lot of recall. It just is. It's just how it's going to be. So that one shouldn't be as taxing on a lot of us. So focus on units 1, 2, and 3. You think you know unit 1. You might think you know the difference between a prokaryote and an eukaryote. But if I'm asking you questions like, oh, if you saw the pathogen had a nucleus, and you weren't able to tell me that it couldn't be bacteria, and you didn't know the difference between prokaryotes and eukaryotes, that was like a chapter freaking like 1 concept. It's in the first chapter. Yes. And that stuff is stuff that we've carried on throughout. So there's stuff that you guys will know, like gram positive versus gram negative. Well, don't focus on that stuff when you're studying, if you already know it. Focus on the stuff you didn't know. Well, if you go back to your notes, you look over those. Oh, yeah, there's something about, oh, yeah, that eukaryotes and prokaryotes, or eukaryotes and eukaryotes. You know, what's the difference between those? You know, make sure that you're looking through those notes and you still can recall those concepts on the ones that you can't explain back to somebody verbally. If they were asking you questions about it, that's how you need to be thinking about your notes, right? If you get to things like that, if you can't explain or are difficult to you, make a note of it. Those are the things you need to be crunch timing with. Focus on the stuff you know you suck at. Then go back and work on the things you suck less at, and then less at, and then you can go and review the easy stuff.
How many microscope 5 questions in the exam?
I don't know the exact number
We had
off the top of my head. We had 9 in the first one. It won't be that many. I think it's like 6 or 7 maybe, but it'll be close. Because there's going to be questions about the parts of the microscope. Again, it'll be a different part, by the way, just FYI. And its function. There'll be at least 1 gram stain slide, at least There'll be capsule, there will be acid fast, there will be endospore in some form. We'll have questions about those things on there. There will be a basic like simple stain sort of question, like cell shape or arrangement, those sorts of things that might come up. That was something that we saw on the midterm exam too, but it'll be different versions of those similar kinds of questions.
But would it still be like, what kind of stain is this thing?
Yes.
All righty.
I expect you to know the stains just as well as you did for the first exam. The stains. I will not be testing you on ELISA, any DNA stuff, including transformation, or Kirby-Bauer. Won't be testing on that. So that's what we're kind of going over, is semi-comprehensive. So it will have all the stains on it and the microscope functionality concepts. It will have those things on there, but it won't have Kirby-Bauer. It won't have DNA stuff, no transformation. I will ask you probably a question about the quad-streak method, and it's probably going to be the same question. There's so many things I can ask. There's only so many things I can ask about the quad-streak method and what it's used for. It's for isolation. And which quadrant had the most isolated single colonies? 4. So that'll be a gimme on that. But that's 2 questions. You still got 48 other questions that'll be on there. The things that trip people up the most on the lab final. You might think it's going to be the colors and the recognition, but you're probably going to work pretty hard on your colors and your pH changes and knowing which one is going to be which. So you might think it's going to be that, but the problem is that it's usually the things that are going to be the concept-based question. So there will be a question on the lab. There will be 2 questions on the lab final, 2 questions, not 2 stations, but 2 questions on the lab final, which will ask you to use a dichotomous key to tell me about your information. So I'll give you something like, okay, it was urease positive. It was, I don't know, indole negative. It was able to hydrolyze gelatin. It was a cocci. It was gram negative. Whatever. And I might say, which organism is it? The question will be which, and I'll just list like a whole bunch of results. It won't be in the chart like yours is. But it's the same exact type of question. So you guys are used to using the chart and like saying, okay, it's asking me about gram negative. Did it have gas and glucose or not? That sort of a thing, and going that way. But people see it listed out in a big old paragraph of like, it was positive for these tests and negative for these tests. And they're trying to find all of those tests that I'm listing as if they all matter. They're not all gonna matter. They didn't matter on your sheet either. When you look on your sheet and it's asking you, did it have gas and glucose or not? Are you necessarily going to need to know if it was indole or gelatin? Maybe not, depending on which route you took. So you only going to need 4 tests that got you to your answer, tops. You're going to have a dichotomous key that I'm going to give you, and I'll either give you a list of results or I'll give you a chart. One question will use the chart just exactly like the ones that you guys have in lab. One of them will just be a list of results. And I tell you, people are just crapping all over that question every semester. I'm literally giving you the test results and a dichotomous key and just asking you what organism it is. But people are seeing that it is gelatin positive. So they're finding the gelatin in the dichotomous key. They're not following the route. I'm giving you more information than you need. So is the chart that you guys are using. It has more information than you need. But some people get lost in the big list of results. Just because I'm giving you all those results doesn't mean that they all mean something. It's the same thing on the lecture questions too. We have a lot of lecture questions that are like that. I'll give you a whole bunch of information, but what might really matter is that it's endospore positive, right? And guaranteed, most of your questions on your nursing exams will be like that. It'll be a whole just crap load of information, and you have to find the like one thing maybe. That's what it's really asking. Which of these could be endospore forming bacteria? But it could be a whole paragraph about crap. Right? Almost, and I mean, when I say most, I mean like all of your questions on your nursing exams are leading up to the NCLEX. They're all going to be written in the way that the NCLEX expects you to know. All of these might be bacteria. This is asking all this information about this bacteria and which of these bacteria falls into the information I'm giving. Right? That's going to be the goal when you guys get in nursing school. If you don't learn how to do those questions now, you will fail out of the first or second semester in nursing. There's a reason why 70 people just failed out of the second semester in nursing. Yes, yes. I'm not spouting out information just out of my butt, guys. Half of the people don't make it through the nursing program. You think you get into nursing and you get to be a nurse? BS all day long. You got to pass the program.
The easiest part is getting in.
They have the highest pass rate in the NCLEX for the people that make it that far. I've said it before, but I'll say it again. For the people that make it that far, 50% of you won't. All the thousands of dollars that you put in your education, if you don't pull up your big girl panties and learn how to learn, will be for nothing. It's hard work. It is hard work, period. It's going to be hard and it's going to be overwhelming, but you've got to learn to truck through it. You have to, or you won't make it. There'll be thousands of dollars down the drain, and who do you have to blame for that? Not me.
I almost pointed at you.
Yeah, it's yourself, right? Yeah. So this is the time. This is the time. I want you guys to learn how to learn and pass my class so that you can pass your classes in nursing. But you've got to learn how to learn to do that. This is not your hurdle to jump. This is not your hurdle. And when I tell you this, you talk to any of the people that are actually in like through the first semester of nursing that have actually made it past the first, second semesters. You talk to those people and ask them if they ever use any microbiology information. Ask them that. People that are actually doing well.
Buddy fishes like every day.
Get it out of your head that you're not going to use microbiology. You won't do a gram stain. But if you think you're not going to talk about C. diff or MRSA or the fact that they are bacteria versus fungi, get it out of your head that you're not going to use. That's delusional. It's like saying you're not going to use physiology. I'm just saying you have to learn this stuff well enough that the concepts are foundational, because microbiology and chemistry and physio are foundational for all of your nursing courses. And you're not going to do well, and anatomy too, right? Anatomy, if you learn your root words in anatomy, you'll be okay. Yeah. All right. That's our come to Jesus talk for the morning. Are we ready for the Kahoot for chapter 20? All right. Let's do it. Chapter 20, review for Kahoot. You're going to be fine. You just got to put in the effort to pass this course. And once you do that, you'll learn how to learn. What sort of organisms make up the normal biota of the nervous system? Normal biota of the nervous system. All right, so the answer for this one is none of these. The nervous system and the circulatory system and the lymphatic system do not have a biome. They are basically sterile when it comes to microbes. They don't have a natural biome in them. There shouldn't be no microbes.
Because they are like really sensitive.
Well, just because they don't have a biome, they just don't have one. And it's sterile. It's just one of those contained closed systems. But that means it's a whole stadium of empty seats too. All right, what is the term that refers to the significantly decreased and compartmentalized immune response in the nervous system? It's applied to the eye as well.
I had my doctor's appointment Tuesday, and he talked exactly about that because I have 60% of space in my eyeball.
Oh yeah,
Which is, I'm a major concern with glaucoma.
right. That has a lot to do with the shape of your eye and how the lens, yeah. All right, so when we have this effect of having basically it's a decreased and compartmentalized immune response in the nervous system to protect from the inflammatory response involved in immune response. This is referring to immune privilege. That's the word for this one. All right, that's a term one. This one, select all that apply. Which types of organisms can cause meningitis? There are a lot of different causes of meningitis. Meningitis can even be caused by things that are not microbes, be trauma, things like that. We talked about the microbe causes. There are 3 major versions, FYI. Alrighty, so this is going to be bacteria, fungi, and viruses. We learned about them in that order. The bacterial guys were our Neisseria meningitis, the gram-negative diplococcus, the meningococcus, the worst one. Streptococcus pneumoniae, which is the gram-positive, it's in chains, that one is. Then we had Haemophilus influenzae, that one was a gram-negative. Coccobacillus. And then we had Listeria monocytogenes. Those were all of our bacteria. This is the gram-positive Coccobacillus. Those were all of our bacterial ones we talked about. Then we talked about 2 different fungal ones. And then we talked about viruses. Most meningitis is going to be caused by viruses. 4 out of 5 cases of meningitis are going to be caused by viral. And most people are going to clear out viral meningitis. It's usually associated with enteroviruses or herpes virus.
Aren't prions just misfolded protein?
Yes, prions are just misfolded protein. They are not associated with meningitis. They are associated with subacute encephalitis. We call the disease that prions cause, we call that spongiform encephalopathy. Pathy means disease, encephalo is brain, and it's spongiform because of the holes that it forms in the brain. All righty, next question. Which pathogen causes the deadliest form of meningitis? So I did just say it. Let's see if you were paying attention. These are all bacteria. 3 of them we talked about being associated with meningitis. Which one causes the deadliest version? All right, this is Neisseria meningitidis. This is the meningococcus. Meningococcal meningitis is the deadliest version. It's the one that leads to the petechial rash and could potentially lead to meningococcemia, which is a blood infection of the bacteria. All right, what type of illness does Naegleria fowleri cause? These questions are very similar to like what you will see on the test for Unit 4, by the way. All righty, so Naegleria fowleri is associated with PAM, which is primary amoebic meningoencephalitis. Do you guys remember what causes the GAM one? What organism that was? Starts with an A.
Yeah, I need to find it.
There, so we're getting there, right? Acanthamoeba, yes, Acanthamoeba. Right, great job.
I needed to highlight more. That's okay.
All right, next question. What type of pathogen typically causes encephalitis? From these choices. Encephalitis for this one. Typically associated with it. Alrighty, so our answer here is arbovirus. We've got some disappointed people out there. All right, so it's arbovirus. These are the viruses that are transmitted by biting insects. Very commonly associated with acute encephalitis. The gram-positive, gram-negative bacteria, I'm not saying they can't cause encephalitis, but more commonly encephalitis is associated with viral infection. Enteroviruses are associated with more commonly with meningitis, not as much with encephalitis. Okay. Righty. True or false? Thanks to the vaccine, polio has been eradicated from the planet.
I don't feel that's true. It's true.
There's a reason that this picture is here, by the way. I'm just gonna hand it back.
There's like one place that still has it, and I picked the wrong one.
Right, so this one is false. We still have polio on the planet, but it's primarily seen in Pakistan and Afghanistan. We haven't been able to get to the rural areas of that, and in war-torn areas, we haven't been able to get the vaccines out to those areas, but otherwise, we're pretty close to it, and we'll probably see that happening in our lifetime. Question A, what kind of pathogen causes Creutzfeldt-Jakob disease? Also known as CJD. Creutzfeldt-Jakob. Okay, so this is a prion disease. Creutzfeldt-Jakob is a spongiform encephalitis caused by misfolded protein in people. So one of the other prion diseases that we didn't talk about in class is chronic wasting disease in deer. So deer have a prion disease. And if you go hunting for deer, you're supposed to be sending samples to get tested for chronic wasting disease. Now, chronic wasting disease, again, a prion disease, is pretty rampant in deer populations, and it was primarily focused in like Colorado, but we have seen a few cases pop up just randomly in Oklahoma, and it might become a bigger issue. Yes.
One time we had a deer with clotting that disease at our house.
Yeah.
And it
And
was-
so usually they're gonna be slower, they might have neurological symptoms, but they can also harbor the disease without any symptoms potentially. No. Up to the hunter to do it, yes. It's actually required by law in Colorado, and you're supposed to be doing it. It is on the hunter to be doing that. Right now, the variation of chronic wasting disease that exists in the deer population has started to spread to other mammals. It hasn't spread to humans yet, but if it
Yeah.
continues, it could potentially. So we got to keep that in mind, that keep it under control in the deer so it doesn't get to the people.
You sign something when you get your hunting license saying you will do it.
Yes.
So that's like left on the hunter's part.
Yes. FYI, you've been reminded. All right, what is the mortality rate for rabies if it is left untreated? Yeah, y'all heard me say this a million times.
20, there's 1%. It's like 80 get tricked. You're gonna dodge you maybe. It'll be fun.
Yeah, so it's 100%. You'll be fine because you'll be dead. You'll know the difference. Yeah. Alrighty. Last question for chapter 20. Which toxin causes spastic paralysis? This was our last two diseases that we talked about. Right, so this is tetanospasmin. So tetanus is associated with a spastic paralysis, where the muscles can't relax. So they're constantly being contracted. If we talk about flaccid paralysis is where you can't contract, like the muscles are just completely and totally inert. That is what is caused by botulinum toxin.
Do you know if they still lobotomize me?
Yeah, usually chemical lobotomization, I think.
I have a sticker, not a sticker, magnet on my car that says on my way to get a lobotomy. Do these lobotomies have like vans?
Yeah.
People forget that JFK's sister had a lobotomy.
Yeah.
Dude, it destroyed her.
Have you?
It's like you're not depressed anymore, but also you don't really know.
You're a vegetable. Yeah.
I want a lobotomy though.
All righty. So, good job with this, guys. We're gonna jump into chapter 21 now. Maybe if I can open it. All right, chapter 21, we're going to be talking about diseases of the cardiovascular and lymphatic systems. Again, I'm not necessarily going to test you on the anatomy of these systems. Well, you'll get tested a little bit on the lymphatic because we talked about that in unit 3, but that'll be a unit 3 issue. So we know the circulatory system, this is a closed system. Your blood stays in your cardiovascular system. It shouldn't be the blood leaving that system. And then the gas exchange can happen across the capillaries, but that these blood cells and the liquid in your blood we consider this to be a closed system. The heart pumps the blood. Blood that's moving away from the heart is called arteries. It's carried by vessels that we call arteries, and blood coming back to the heart is carried by vessels that we call veins. And then the capillaries are where we'll have that crossover happening in the tissues. So this will provide oxygen and nutrients throughout the body, crossing over at those thinner capillary levels in the tissues. Cool. Lymphatic system, we've talked about this previously, but we have the lymphatic vessels that are going to carry fluid from the extremities back towards the heart via skeletal movement. The lymph nodes are clusters of tissue in the groin, neck, armpit, and intestines, and they're scattered throughout the body, but that's where we have the largest concentrations of them. And these guys will, basically, when you have a lymphocyte response, the B cells are multiplying in the lymph nodes, and so that's why they swell up the way they do, because cells are like really proliferating and growing. And then the spleen is also considered part of the lymphatic system and the circulatory system. So the spleen is filtering out damaged red blood cells, as well as being a site of filtering as a lymphatic tissue, filtering out the blood itself, not the lymph in that case. Okay, right, so cardiovascular system, we don't have any biome. It should be a sterile environment as far as microbes go. It is highly protected from microbial infection because it is circulating throughout the body. If you have infection in the bloodstream, that there is a risk of it getting to other areas of the body via the blood moving it around. So when we think about the central nervous system, the nervous system overall and the cardiovascular and lymphatic systems. These guys don't have a biome. They're open stadiums. There's nobody sitting in those seats. That's also why we have to have protection measures in place to keep microbes from getting in there, because once they get in there, there's so many stadium seats for them to multiply and spread around in. All right. In the bloodstream, though, thankfully, it's a little bit different. This is not an immune privileged site. This is the place that the immune system moves around in. And so we have high numbers of white blood cells. We have the lymphocytes. We have phagocytes. All that stuff is found in the blood and in the lymph. So we should have a very strong protective response present in the bloodstream, feeding nutrients and everything all throughout the body, including to the nervous system, right? Any time that we have a medical condition involving the blood, we call it emia. Emia is referring to diseases in the bloodstream. We have, for our microbial related diseases of the blood, viremia, viruses in the blood. These are often linked to viruses that can cause meningitis and potentially encephalitis as well, like our arboviruses that we referred to from the mosquitoes. They come and bite you, the virus gets into your bloodstream and then can move throughout the body through the bloodstream. So viremia, viruses in the blood. Fungemia, fungi in the blood. Bacteremia. If you have any bacteria in the blood, we call that bacteremia. If they are flourishing in the bloodstream, any of these microbes actually flourishing and multiplying in the bloodstream, that's called septicemia. So that's the difference between those. So septicemia is when microbes are flourishing and growing and multiplying and causing infection in the bloodstream where there shouldn't be any. And so they're flourishing, and that's septicemia. Bacteremia is any bacteria in the blood. So there's a difference there. All right. We shouldn't have any biota there. We said it. Let's get into some of our diseases. Let's start with COVID. We think of COVID as being a respiratory disease, but COVID actually can infect lots and lots of cells in your body, and it's moved around by the circulatory system. So while you might get exposed to it in the respiratory system, it will move around throughout the entire body. In fact, you've probably heard about whenever there was COVID outbreaks that viruses are found in all sorts of bodily excretion, not just in your lungs, right? You're not just only coughing it out. It's going to be found in the feces and in urine and in saliva and all sorts of things. So COVID-19 is considered a cardiovascular disease. It is caused by the virus SARS-CoV-2, so severe acute respiratory syndrome. CoV is referring to coronavirus 2, major serotype of this coronavirus. So it's a SARS-based coronavirus, and it is like our different type of virus in that realm. Number 2, okay? SARS-CoV-1 caused SARS. So SARS was acute respiratory severe acute respiratory syndrome, primarily seen in Asia and China is where the biggest outbreaks were. And there was a huge concern in like the early 2000s when there was an outbreak of SARS that it would make its way to the United States and that people would become sick and die. And then we got COVID instead. So it's highly believed, and I'm sure it's they're going to talk about it in a second, but some people say, oh, it came from the bat population. Where do we really think it came from? So all evidence points to it probably came from a lab in China that was working on SARS. And then they were doing mutations of SARS to make it more virulent to be used as a biological weapon. And it wasn't contained in the lab and it got out. And that's what COVID is. So all evidence points to that. This is not just me talking about conspiracy theories, but you know, it is what it is. This book was written in 2022. So whatever. But yes, so we think that it came from the SARS virus, but not on virus because there was enough genetic changes. And the disease is a little bit different. So it uses ACE2 host cell proteins. There's these proteins on your cells called ACE2, and it uses those as receptors to get in. And these ACE2 proteins are found on almost every cell in your body. So it can infect pretty much any cell in your body. The COVID virus can. It's usually transmitted via respiratory droplets, but it can be transmitted a lot of different ways outside of that. The different versions and variants of SARS-CoV-2 are cause of COVID. Alpha, Delta, and Omicron, and there's been other ones since this was published, but they have gotten more and more virulent over time. So if I tell you that Delta and Omicron are more infectious, more transmissible, and cause a worse disease overall, worse pathogenesis than the original COVID virus, and we're sitting here talking about how, like, man, COVID was so bad whenever it first came out, right? Like, we're not getting anywhere near as bad as disease. I don't need to tell you why that is, right? We know why that is. Yes. Vaccines. Yes. Okay. All right. Signs and symptoms. You'll have prominent respiratory symptoms, but infection can cause a cytokine storm in some people. It basically triggers the inflammatory response at a high level, and that's where we start seeing that effect of people who were hospitalized. So even healthy people might have cytokine storm triggered by having COVID. The vaccine helps your body reorient the immune response the way that it should be oriented. So we're less likely to induce the cytokine storm. So it has significantly reduced the number of people having cytokine storm related and then eventually death associated with things like shock and stuff like that that can happen as a result of the cytokine storm. And cytokine storm, hyperinflammation in the lungs and other organs and the digestive tract, and that can eventually lead to a shock response and even death in some people. It is also considered a neurotropic virus. It can affect the nervous system potentially, and that can lead to some of the encephalitis symptoms that we talked about in the previous chapter. A third of infections with COVID are going to be asymptomatic. So that's what I brought this up early in this semester, but just reminding you guys, a third of people that technically are carrying around COVID don't have any signs or symptoms at all. And so this is a bigger issue because we'll be spreading around that virus for people that think they're not sick. And so then other people get it and they might have the cytokine storm response. And so that was the bigger issue with that and why we had to have masks and social distancing and things like that. The two different versions of the vaccine that came out, one of them was based on an mRNA, literally just basically injecting you with mRNA. Your cells would translate it, put the viral spike proteins on the surface of your cells, and we'd have an immune response to that. That's the Pfizer and Moderna versions. The Johnson & Johnson version uses an adenovirus that we've hollowed out and basically put the spike proteins of COVID on that. And then you respond to that one. Yes.
But out of the two, what would be better for something cardiovascular?
So I've had people ask me that question before, and I don't know the answer to that one yet. So do your due diligence when you're trying to decide who should get what kind of these vaccines. I don't know off the top of my head. With side effects that are associated with how the body responds to each of these, do your diligence and looking up the actual numbers associated with that in response to whether you think somebody's immunocompromised or might have cardiovascular inflammatory issues already. Do your research on that. Don't just rely on somebody else telling you, oh well, I got it and it did this, right? So look at the numbers for that.
I've been looking at the Johnson.
There are medications as well that can be used to treat COVID, and basically treat that as well as maybe immunoglobulins and people that survived it and stuff like that. Moving on, endocarditis. Cardio is the heart and itis is inflammation. So the inflammation of the inner lining of the heart here. So this is caused usually in our case, because it's microbiology. This was the version that they're caused by infection. When you have this, you can have subacute or acute versions of these. We have damage to the heart valves as well as the tissues in the inside of the heart as a result of the inflammatory response to these and usually two bacteria that might be causing this. For acute endocarditis, we will have fever, anemia, abnormal heartbeat, heart attack, shortness of breath, chills. You might have abdominal or side pain, usually associated with the spleen dealing with the inflammatory effects in the body. Your spleen is like over here, kind of up under your ribs a little bit on your left side. Abdominal or side pain from that. Janeway's lesions and Osler's nodes. This is where if you start having dark spots appearing in your nails, like brownish, kind of, it looks like scabs, and that it is. It's from blood, from burst blood vessels, and you'll start seeing that appear in the nails. Those are Janeway's lesions, and Osler's nodes are going to be where we start seeing those spots appearing in like your calloused areas of the skin, maybe in the palms of the hand or the soles of the feet and things like that. Again, usually from ruptured blood vessels associated with those. If you have subacute endocarditis, it's similar. It's going to be less pronounced than the acute, and it's going to be a longer term, usually more chronic. And it will usually over time, because you have it over a long period of time, you'll have more morphological effects appearing in your patients. They'll have an enlarged spleen from dealing with the inflammation from this, as well as clubbed fingers and toes. So if you guys don't know what a clubbed finger or toe is, where like the nail, instead of being more flat, like maybe my nail is, it starts to round over and it has like a rounded appearance on it. And also you might have a little bit of enlargement of the nail bed as well. So that's clubbed fingers. You should not have that. That's not normal, ever. Okay, so if somebody has clubbed fingers or toes, that is usually a sign of something is wrong in the body. So that could be due to nutrient deficiencies, genetic defects, or endocarditis. So if somebody has clubbed fingers or toes, there is a reason for that. It is not because you have that shape of a finger or toe. Yeah, so it'll like literally have a rounded appearance to it. If you've seen it, you know what I'm talking about.
Yeah.
So it doesn't look like some serious abnormal effect, but it is a sign of underlying disease. All right, sepsis. Again, septicemia, where you have flourishing of microbes in the bloodstream or the lymph. This is signs and symptoms will be fever, altered mental state, shaking, chills, gastrointestinal symptoms. This will be systemic effects, basically, because you had microbes floating around your bloodstream where there should be no microbes. And the inflammatory response is getting spread throughout your entire body through the bloodstream. And so this is a systemic inflammatory response to bacteria and sometimes fungi, but it's usually bacterial in nature when we have this happening. Endotoxic shock is the biggest risk associated with septicemia. Endotoxin found on gram negatives. This acts as a toxin that stimulates the inflammatory response. It will lead to a drastic loss of blood pressure and can, even if it continues, leads to death and coma associated with just the inflammatory response driving this whole thing. This is usually going to be associated with gram-negative endotoxin. Gram-positives have certain molecules that can also trigger a similar effect. In fact, when we talk about toxic shock syndrome with tampon use, as we talked about previously, that's usually caused by a gram-positive organism. And so those guys have a different molecule in them. Remember, even peptidoglycan is a PAMP that can serve as a way to initiate the shock response, your inflammatory response. But it's usually going to be other toxins that those microbes make that will lead to a similar effect. Let's move on. The plague. There are 3 different manifestations of plague: pneumonic, bubonic, and septicemic. We're usually used to talking about bubonic plague. You might even call it the Black Plague. The Black Plague is the septicemic version, right? Bubonic plague, where you have buboes. A bubo is an infected lymph node. So if the lymph node get infected with this bacteria, this is a bacterial disease. The bacteria are transmitted by the bites of fleas. So again, bacteria that are transmitted by a biting insect. And the rat populations in the Middle Ages, the rat populations were maintaining the bacteria. The fleas would bite people and transmit the bacteria to the people. And so bacteria get into the bloodstream and get into the lymphatic system, and then they get able to populate within the lymph nodes. We get swollen, infected lymph nodes that you can actually see and palpate from the surface of the skin. And they might even be black or dark red in nature, and they might rupture and ooze pus and things like that. Those are buboes, and that's bubonic plague. The bubonic plague is the tamest version. And we're talking about a pretty high mortality, or about 20% mortality rate for bubonic plague. Pneumonic plague is the next worst version of it. Pneumonic plague is actually transmissible between people. So if somebody has pneumonic plague, if it's moved into the lungs of a person, they can actually cough and spread that to other people. This is the only one that is transmissible between people, typically. The other one is septicemic. This is when we have the actual blood infection throughout the entire bloodstream. And it's not just localized to the lymph nodes. If it gets throughout the entire bloodstream, septicemic plague will happen. We have the inflammatory response happening again. This is caused by bacteria. The bacteria is called Yersinia pestis, and Yersinia pestis is a gram-negative bacteria. You have an endotoxin response going on. And so what will happen is the endotoxin response going onto the bacteria, as well as the toxins the bacteria produce, in the extremities and in the capillaries, will start to have damage to the blood vessels, and that will lead to necrosis. And then that will lead to fingertips and toes and extremities turning black. That's why it's called the Black Death, because of the necrosis of the extremities associated with it. The Black Death, or some septicemic version of the plague in the bloodstream, is 30 to 50% mortality if we treat you with everything we have in our arsenal. So even if I treat you with everything we have, if you have the septicemic version of the plague, you're looking at a 30 to 50% mortality rate with treatment. If you don't get treatment, this is a 100% fatality rate here. So this is some images of some buboes. Again, it's caused by a bacteria called Yersinia pestis. There's some images of it in the next picture next to that. Where do we have the plague today? Well, it's still present all over the world. Maintained in animal populations and rodent populations. In the United States, this is a map of outbreaks since 1970 to 2017. This is every single outbreak that there was. So this is not a super common disease that we have in humans, but it's very rampant in prairie dog populations in the Four Corners region of the United States. So prairie dogs do harbor the plague. They might be cute to you, but maybe don't get near them because their fleas will bite you and you can get the plague. Okay. So we have some outbreaks here, primarily in the Four Corners region. That's Utah, Colorado, New Mexico, and Arizona area.
Can't trust the Mormons.
Yeah. All right, moving on to mono, infectious mononucleosis. This is caused by the Epstein-Barr virus. The virus will primarily multiply in the lymph node tissues. So again, we will have development of swollen lymph nodes And lymphadenopathy is a defined feature of mononucleosis. So our mononuclear cells are what are being infected here, and this will include your lymphocytes. And so the lymphocytes will get infected and then hang out in the lymph nodes. Lymph nodes get swollen, and that. More than 90% of the world's population has been infected with EBV at this point. So you have had it, probably. Okay. Even if you don't remember having mono and the signs and symptoms of it, most of the time when children get it, they're asymptomatic when children get it. But as you get older, you get into our teenage years, your immune system isn't quite as strong as it was whenever you were younger. And so you're more likely to, as you get teenage and young adult years, as you're getting exposed to mono, then you will have outbreak of actual disease associated with that. It is a herpes virus. So once you have it forever. And you can have resurgence of mono at any point in your life. And some people just do their immune response working the way that it does. Will actually have resurgence of mono like regularly. I had a student in the previous semester who his dad would get mono like every year. He would just come out, he'd get mono. He didn't get it from people, he got it from his own body, just activating or whatever. So these guys are viruses that live in the lymphatic cells, and they incorporate themselves into the host DNA. And so it becomes a permanent infection after that. Infection during the teen years results in disease about 30 to 77% of the time, depending on the populations that we're looking at. So again, a lot of people can have this disease and not have signs or symptoms associated with it.
It's also called the kissing disease.
It's also called kissing disease. It's usually spread through saliva. It's a pretty contagious illness. And if you've had it before, then you know it's horrible. You have signs and symptoms. You'll get severe fever. You'll get shakes and chills. It's much worse than the flu. And it usually can last for about a month even. Most people don't recover from it for about a month. So it's pretty severe. Yes. Yeah, not uncommon. Yeah. Some people, it takes a long time for them to recover. I say a month, like that's like if you are very healthy and your immune system's responding properly. Other people can have it. You can have long-term effects from mono that can last a lifetime. So some people can even have that happening, but we won't get into that stuff right now, but we'll talk about the general idea of it. Moving on to anthrax. We talked about cutaneous anthrax when we talked about the skin diseases, but anthrax can also infect the bloodstream. And if you do, it's going to be a much worse outcome if you have it in the bloodstream than if you just have a skin infection with it. Again, it's a zoonotic disease that is present naturally in herbivore populations, usually in livestock is where we see that. On the skin, it's cutaneous. In the lungs, it's pulmonary. In the GI, it's gastrointestinal. And in the nervous system, we call it anthrax meningitis. So it can infect the bloodstream as well, which is the septicemic anthrax. Anthrax is caused by the bacteria Bacillus anthracis. So spore-forming bacteria. So we already know that because it's called bacillus. Remember, if it's called bacillus or clostridium, it makes endospores. And if it makes endospores, it is gram-positive. So these are the things that we already know about it based on what we learned in chapter 4. These guys are aerobic. So that's the difference between bacillus and clostridium. Clostridium is anaerobic. And so these guys are aerobic. They're also catalase positive, so it's a way that we could identify them as well. They have toxins that they make that I'm not going to test you on. Moving on to the hemorrhagic fever. Hemorrhagic fever is defined basically by fragility of the blood vessels. As the body is responding to the virus or the bacteria that causes the hemorrhagic fever, the inflammation response will cause the fragility, literally like fragile blood vessels, and the capillaries can start to burst. So these guys might lead to petechial rashes, but eventually over time, due to the pathogenesis of the virus, it will cause degradation of the vasculatory system overall, and you will even see like weeping of blood, oozing of blood out of orifices, and things like that. So hemorrhagic fever is defined by the fact that you have hemorrhaging going on. So this is leakage of the blood vessels and blood into the tissues where they wouldn't normally be leaking into. Most of these guys, most of hemorrhagic fever diseases are caused by RNA enveloped viruses. And a lot of them are distributed by Aedes mosquitoes. So we have Aedes mosquitoes. We've already talked about the Aedes guys in the previous chapter with our arboviruses. And we have more widespread of the mosquito population as we have global warming. And so more and more of those mosquito populations that carry these diseases have moved further and further north. So that now some of these diseases that are hemorrhagic fevers that we didn't used to see in the United States are now becoming endemic, especially in the southern United States. We're talking Texas and Florida. These are the areas where we will start to see some of these, okay? So here are hemorrhagic fevers. Yellow fever, It's usually associated in Africa and South America. Dengue fever. Dengue was originally mostly localized to Asia, Africa, and South and Central America, so the tropical regions of the world. Now that we have started having warming of the climate due to the climate change going on, whatever the cause of it, whatever. But global warming is causing the mosquito populations to move north. So we now have dengue fever becoming endemic in Texas. And so dengue is a possible hemorrhagic. This is what we call dengue causes a breakbone fever. So you'll have like such strong chills and stuff associated with this that technically could break bones if you were a fragile person. And that's how strong the chills can be associated with dengue fever. Chikungunya is a viral disease that is now endemic here in the United States as well. And it doesn't usually cause hemorrhagic effects, but it can be associated with hemorrhagic fever. It's usually associated more with neurological side effects. It is here in the United States though. Then we have the other guys that are not spread by biting insects. So those three here, yellow fever, dengue, and chikungunya, they're all transmitted by mosquitoes. But we know Ebola is not transmitted by mosquitoes, right? Where this is like blood to blood contact, typically associated with Ebola. Ebola is not airborne in humans. It is airborne. There is a strain that is airborne in primates. So far, it cannot infect humans. And it is fairly deadly in primates. So we're talking about 60% mortality rate for the Ebola restin virus. But the other ones, Ebola, Zaire, and Ebola, Ivory Coast, and all the other strains of Ebola that we have out there, we're talking like 90% fatality rate. Super high. But at the same time, we usually have a limiting outbreak there. Most of the time, once it gets to developed countries that are educated about hygiene and things like that, you're not going around and rubbing yourself all in the blood of somebody that's bleeding out. We have learned better. So we can usually contain the outbreaks fairly well once it gets outside of the source of where the outbreak started, which is typically going to be in Africa, in the tribes of Africa that rely on bushmeat for survival. So they're hunting animals to eat, whether it's primates or bats or other animals that could potentially be infected with Ebola. And then they get infected with it, and then it gets widespread outside of those outbreaks. Okay. We think that bats are the reservoir for both Ebola and Marburg. Marburg is very similar to Ebola. That's really all you need to know about it. But the first outbreak of Marburg happened in Marburg, Germany. It was from imports of, I think, animals from Africa going through quarantine, and it was just discovered in Marburg, Germany. Ebola restin, the one that I was just talking about, was discovered in Reston, Virginia, here in America, and that was found in primates. That's the airborne version. And that for some reason, the monkeys that they were bringing in for testing at USAMRIID, which is the Army Research Institute for Diseases, the monkeys there were just dying, and they didn't know why, because they were kept in like different rooms, but it was spreading through the ventilation system. And it was Ebola just spreading throughout all the monkeys that they had brought in for testing. And then they had to like quarantine the people and like make sure that people weren't getting it. And so far it's confined to primates and not human primates, but like non-human primates. Anyway, but that exists. That's the thing. So to think that it couldn't be airborne would, I think, be a little bit naive. So we just got to make sure we're containing outbreaks so that there is less likelihood of it mutating and moving into the human populations. But anyways, there's a whole book on that. If you're interested, it's called The Hot Zone. I read it, and that's the whole reason why I went into studying hemorrhagic fevers in graduate school. That was my specialty. So Lassa fever, this is a viral infection that's usually asymptomatic in 80% of human cases, but in the 20% of people that do get Lassa fever, we're talking about extremely severe hemorrhagic symptoms. So then we have some fevers that are non-hemorrhagic. So they are fevers caused typically by microbes that are transmitted by biting insects. Not all of them are, but they're very commonly associated with biting insects. So we have fever. They are associated with the cardiovascular system, transporting these guys throughout the body. So you'll have fever development, but you won't have that very fragile vasculatory system effect that we see in the hemorrhagic fevers. Some of these guys toe the line a little bit, though. They might have a particular rash associated with them, but you won't have a foam bleeding out associated with that hemorrhage. All right. Most of these are bacterial that we're going to talk about, okay? So these are most of them bacterial, but babesiosis is one of them that's protozoal, but all the others are going to be bacterial. All right, Lyme disease first. We know it's transmitted by tick bite. And we have development of these very typical bullseye type of rashes associated with where the tick bite was. And that's usually a sign that you maybe have Lyme disease. If you don't have the bullseye rash, we're gonna be less likely to think that it would be Lyme disease, but you can still get Lyme disease without the bullseye rash. It's uncommon. The signs and symptoms of Lyme disease, this is a fever, headache, stiff neck, and dizziness that can eventually lead to neurological symptoms whenever the bacteria moves into the nervous system. This is a very slow progress disease, and the bacteria that cause Lyme disease are, they live inside of the cells, and so they migrate through the cells that way. Yes.
So I was diagnosed with Lyme disease in 4th grade, but I had no bullseye.
Yeah.
For one of my first early, we caught it really early.
Mhm.
I had ball palsy.
Yes, so neurological, we're talking about severe neurological symptoms usually that develop from these people. It is treatable with antibiotics, but it's usually like a longer course. It's not like your 10-day antibiotics, right? This is something that lives inside of the cells, and so we have to have a longer term treatment involved in these guys. And it's hard to eliminate it completely. So sometimes whenever we treat this, people can still come back with it later on because we didn't eliminate all the bacteria with it. It's caused by bacteria called Borrelia burgdorferi. They have these spiral shapes, and they have little hooks at the end that help them get into the cells and attach to the host cells. It's rough, man. You don't want it. Lyme disease, again, it was discovered in Lyme, Connecticut, old Lyme, Connecticut. Anyways, yeah, it's named after the place. Anyway, so these guys will bite, it lives in animals and then can transmit to people. This is not something we have in Oklahoma. It's found in the northeastern United States typically. That doesn't mean you can't find cases outside of Oklahoma. It's just less common. Ehrlichiosis and anaplasmosis. This is something that they will ask you questions about if you donate blood. So these guys are bacterial diseases. Ehrlichiosis is caused by Ehrlichia chiefensis, and then anaplasmosis is caused by Anaplasma phagocytophilium. Again, you don't have to write them, right? So these guys are transmitted by Ixodes ticks. And so Ixodes ticks are hard shell ticks. So you have this ticks that are like big gray ones that are soft, and they fill up with the, I hate ticks, you guys. I hate ticks. But the hard shell ticks are the Ixodes ticks. And so these diseases are associated with the hard shell ticks. Anyways, you'll have acute febrile state. So you feel like crap. That's what that means. You feel like crap, and you have these symptoms of headache, muscle pain, and rigors associated with that. And most people will recover naturally and get over it on their own. Babesiosis is protozoan. It's our one protozoa that we'll have in this category. It's carried by ticks and will be treated with a combination of medications that can be used to treat protozoa. We'll also usually has similar symptoms to Ehrlichia and Anaplasma. So if you are having symptoms that is associated with a tick bite, we will often treat with antiprotozoal and antibacterial guys to help treat that. Overall, it's probably babesiosis or Ehrlichia or Anaplasma. Whoops. Okay, yes, Rocky Mountain spotted fever. Rickettsioses are diseases that are associated with Rickettsia bacteria. The most well-known one is Rocky Mountain spotted fever. So Rocky Mountain spotted fever is a rickettsial disease. Rocky Mountain spotted fever is caused by bacteria, Rickettsia rickettsii. So easy, same word there. So it is a bacteria that is transmitted by hard ticks, the Ixodes ticks again, and it is found throughout the United States. You will get chills, fever, headache, and muscular pain. Then you will have a spotted rash. It's associated with the petechial rash there. So this one is the one that toes the line of hemorrhagic. Okay, so you get the rash, it is petechial rash, and you will eventually, some people will get cardiovascular disruption, including hypotension, thrombosis, and potentially hemorrhage in some cases. It is a 20% mortality rate if you don't treat it. It's still 5 to 10% mortality rate with treatment. I had a student that was a paramedic that went to a rural location and got a tick bite and developed Rocky Mountain spotted fever as a result of that. She was bedridden for months. This is a disease that you don't want to get. It's not like an easy thing to recover from.
My husband had it and was diagnosed for a very long time.
Yeah,
And
yeah.
he was bedridden for almost 7 months.
It is rough.
Not like, but he just had zero, and he had so many problems. From
Yeah, from the inflammation, and it's just like taking so much out of your body, trying to keep all that up over a
it.
long
They
term.
misdiagnosed it so many times.
Yeah, and that's one of those things. It's like it's rough enough to have that sort of chronic underlying affection, and then some people also have that issue where they're going to have the hemorrhage and thrombosis and things like that. Like, so this is not something to be taken lightly. If you have a tick bite, monitor your signs and symptoms afterwards. And if you think that it is Rocky Mountain spotted fever, go to get the treatment. And again, still 5 to 10% mortality rate is still pretty high. So it's nothing you want to play with. Again, this is the rash that is associated with Rocky Mountain spotted fever. You do have a rash developed from this. All right, moving on to Chagas disease. Chagas disease is a protozoal disease transmitted by this biting bug called what we call it, has it a name? We call it like the assassin bug is what we call it here in the United States. But these guys are biting insect. They look like this. So you might have seen in your garden some bugs that look like this that have a big old like wheel shape on their back. Those are wheel bugs. Those are fine. Those don't bite. These guys though are associated with basically living in houses in the United States, by the way. Even here in Oklahoma, we have this. So this is a biting insect that lives in houses, and they're about this big. They're called triatome bugs or triatomine bugs, kissing bugs or assassin bugs also. Kissing bugs is usually what they're called, but they look like this, exactly like this. And they live in your houses. And they like to live and nest in like the insulation in houses or in your attics and things like that. You might see them outside as well. But they are very common insect in Oklahoma. I mean, very common. Just because you haven't seen them doesn't mean they're not there. You don't usually see them. And every apartment that I've lived in, I've found at least one. So these guys will bite, and then they like lap up the blood. They don't like use like mosquitoes will use a proboscis and inject into like poke into your blood. These guys don't do that. They bite into your skin and like lap up the blood. And then when they are feeding, they defecate. And then when they defecate, the protozoa will get defecated out. And then those protozoa can enter into the skin from there. Where they enter into the skin when this feeding is happening, it gets an inflamed area that we call a chagoma. It's associated with Chagas disease. The protozoa were able to get into the body that way. So this is the biting one associated with Chagas disease. This protozoa is called Trypanosoma cruzi. So if you have ever donated blood, they will test you for Chagas disease. Yes, technically they'll test you once, and if you don't have it, they're going to assume that you don't have it, because usually this is something that's transmitted to babies. So like they'll crawl into the crib and like bite the babies. No, people that are adults, you'll get woken up by that and they'll like swat away the bug or whatever. But babies are more at risk, and little toddlers are more at risk for this sort of a disease. Again, not super common, more common in warmer climates, but we do have this here in Oklahoma, so do be aware of this. So this can lead to an acute or a chronic version of the disease. If you have the acute version, you'll have a fever, nausea, fatigue. You'll have that swelling area where the protozoa got into the body. It leads to development of a chagoma. It's usually near the eyes or the mouth that you will see these bite sites. Then the chronic version of this, you'll have inflammation and disruption of the heart, brain, and intestinal tract. And this can even lead to death as a result of that. So this is something that can be pretty serious as well. So we've seen trypanosoma in our lab. Remember, this guy was on your test. So this is Trypanosoma cruzi. The one that we saw in the lab was Trypanosoma gambiense, and that's associated with sleeping sickness. It's a very similar microbe, and that leads to sleeping sickness. So people will just fall asleep while they're driving their cars, and eventually can lead to the heart essentially like failing. So we call it exploding heart syndrome. But like a lot of the inflammation associated with the heart causes so much damage that they like will just die from that. This is related to that. So this is the trypanosome in the bloodstream. It's the little squirrely snake looking guy hanging around all the blood cells. All right, moving on to malaria. Malaria is another protozoal blood disease. It's transmitted by mosquitoes. So the protozoa get in, like basically regurgitated into the host, the people typically, and then will multiply in the bloodstream, in the liver cells, and places like that. Signs and symptoms cycle over 48 to 72-hour intervals, and that's usually associated with the life cycle of the protozoa in the bloodstream. So they'll live inside of the blood cells and multiply, and then they'll all rupture out of the blood cells, and that's when you start having the symptoms associated with malaria. Chills, fever, and sweating are common with malaria. Falciparum malaria, that's caused by the organism Plasmodium falciparum, has a more severe symptom presentation. So we have persistent fever, cough, and weakness for weeks. And so they're also having these cycles of the chills, fever, and sweating. And when they have like their sort of chills, we have like a serious like muscle involvement involved in that as well. This can move into the brain as well as leading to neurological issues. So these are caused by Plasmodium species of AP complexans. So AP complexans are a type of protozoa. Remember how protozoa can move 3 different ways or be non-modal. So they can either have flagella, they can be amoebas, or they can have cilia. And if they are non-modal, they are apicomplexans. And so apicomplexans are the non-modal guys, and so they're just being moved around by their environment. And all of them are parasitic. And so malaria is one of them. These guys are named Plasmodium something. The worst and most common one is Plasmodium falciparum. And all of these other versions can be found in different areas in the world. It's not just found in Africa, sub-Saharan Africa, it's found throughout the world. We've had a few cases of people having malaria in Florida lately. And we think that it's related to travel to other areas, but we have the mosquitoes that can maintain malaria in the United States. So if it gets moved, if somebody that has malaria comes to the United States, gets bitten by one of our mosquitoes, the mosquito becomes infected and then can spread it around to other people. You get what I'm saying? So there's a risk there for it to become endemic in the United States. We have all of the foundation for that to happen. Malaria is treatable with protozoal drugs, but it is rough, man. Those drugs are rough on your body, and treating malaria is rough. Having malaria is rough. It is not a disease you want to have. We tried to develop vaccines for it, but they have the ability to change their antigens on their surfaces. So the vaccines just can't keep up with that. Vaccines haven't been super effective either, but there are vaccines available, but you need multiple boosters for it. And like if you lived in sub-Saharan Africa, what are the likelihood that you're going to go for your boosters? All right, so that's the issue. So really dealing with malaria is more or less targeted to controlling mosquitoes. And so here are the things, obvious things, I feel like, in dealing with mosquito populations: eliminating standing water, using insecticides. We have genetically modified male mosquitoes that can't reproduce. The female mosquito mates one time. So she'll mate one time with a male, and then if she is able to make eggs, lays the eggs and then dies. So she'll mate one time. If she mates with a male that is sterile, she'll just die. And so we can introduce sterile males into the population to eliminate mosquito populations. Mosquito nets, screens, and repellents are helpful for keeping the mosquitoes away from the people. And we also have human chemoprophylaxis. If you're traveling to areas where malaria is endemic, then we will treat you with quinine-based drugs while you are there so you don't get the malaria. So it's better to prevent it than it is to treat it. Cool. All right, HIV. This is human immunodeficiency virus. It is related to the development of AIDS, which is acquired immunodeficiency syndrome. This leads to opportunistic infections like pneumonia that's caused by pneumocystitis gyrovecii and Kaposi's sarcoma, Burkitt's lymphoma. Again, we talked about Burkitt's lymphoma previously. If you get mono and one of like infectious mononucleosis by Epstein-Barr virus, that everybody we've already said probably has had it, over 90% of the population has. So that virus lives in your mononuclear cells forever, including your B cells, your lymphocytes. And so our CD8 cytotoxic T cells are able to keep them from overgrowing as a result of cancer that can develop from disrupting our cell cycle from those viruses integrating into our DNA. So our CD8 T cells will kill that off. But if you don't have T helper cells, which is the cells that HIV infects, if you don't have T helper cells to help trigger the cytotoxic response, they get Burkitt's lymphoma, which is a cancer that develops from just having had mono. And so things like that, their immune system isn't controlling, that they'll have issues with. The immune system tries the best that it can, but it can't maintain it. AIDS is defined by the number of T helper cells that you have. So we're looking for CD4 positive T cells. And once your number declines past 200 cells per milliliter, that's considered AIDS. We don't define AIDS necessarily anymore by the signs and symptoms so much as we're defining it by your T cell count once you've been diagnosed with HIV. Again, opportunistic infections, immune system deregulation. You don't have proper immune system response to things that other people are able to fight off pretty easily. They'll have weight loss, diarrhea, they'll have maybe anorexia and things associated with just poor health overall. So HIV is a retrovirus. It's able to take its RNA genome, turn it into DNA, and then the DNA integrates into the genome of the cells that it infects. It is a retrovirus in the genus lentivirus, just FYI. There's two major types of HIV, HIV-1 and HIV-2. The more common one is HIV-1. And it's related to simian immunodeficiency viruses in chimpanzees. We think that's actually where it may have mutated from. So what happens is this RNA virus gets into the body. It uses mRNA. It takes the mRNA into the nucleus and it's able to reverse transcribe the mRNA into DNA, which then gets integrated into the genome. The virus can then make viruses from the DNA genome as it needs or as it sees fit. It's transmitted through blood sources, and that's it. So I want to be very clear about that. HIV is transmitted through blood transfer, and that is it. So it can be transmitted through trauma, mixing of blood, needle sharing, sexual transmission, because there are micro tears involved in sexual activity. That's what we think is involved in the blood. It is present in semen, but it's definitely higher concentration in the blood. And then we have, and it has to get into the blood. It has to, like that's a requirement of it. So if there is semen that is contaminated, if you got that contaminated semen on your intact skin, you wouldn't get HIV. It has to get into the bloodstream. So that's where the micro tears come in, or with the friction involved in sexual activity and all that. And it's also present in breast milk as well, but it's less likely to be transmitted that way. We can actually treat mothers with antiretroviral drugs, and you should be able to have a healthy baby that doesn't have HIV nowadays. So urine, tears, sweat, and saliva do not have high enough numbers of the virus for it to be considered transmitted that way. It was originally found in populations that were practicing in gay relationships, and so it was originally called, AIDS was originally called GRID, which was gay-related immunodeficiency. Yes.
So you said if you treat a positive infected mother with the drugs, the baby can.
Right, there won't be enough virus there for it to cross into the
But
baby.
at what point does the mother need to seek treatment, from the very beginning or third trimester?
I'm
or
hopefully,
5
so
days?
yes, that's a good question. And I'm sure there are studies that have been done on that, but I don't know necessarily the answer for that. But most of the time people aren't finding out that they have HIV in the middle of their pregnancy. If they did, we're going to start them on retrovirals and like cross our fingers. And that's pretty much what we can do. But pretty much like from conception, there's a risk of it being transmitted through the baby through the placenta. If we're not on retrovirals. It's actually pretty high incidence of having a baby that has HIV if you are not on retrovirals.
But you normally know beforehand,
Yeah,
right?
most people do. Most people do, but like you said, like there's going to be cases where they wouldn't.
There's gonna be people that never went to the doctor.
Yeah,
and
and
tried to
so-
figure it out.
Right, and so that we wouldn't know, and then that baby would have HIV and would have to be put. Hopefully, that baby would be put on retrovirals after that, and we can prevent like the immune system from being attacked that way. But it's hard. So we thought it was a gay-related disease because people were ignorant, but we have found out that this is just a human disease. 50. Get this, y'all. South Africa, 50% of the adult population has HIV. 50%. If you think HIV is something small that isn't growing, it's still out there and it's still a problem, even in developed countries. Because I mean, South Africa, like good portions of their cities are considered developed. And so those are the areas that are like, we're talking Cape Town and places like that. So 50% HIV positive. So that is something. United States, 15% of the people who have HIV in the United States don't know they have it. So, you know, bag up, folks, like use the condoms, use the protection and everything like that. Somebody that has HIV, once they test positive for HIV, you have it lifelong. It's something that you don't get rid of. Having HIV is not the same thing as having AIDS. AIDS is determined by your T cell count and the opportunistic infections that come along with that. You can be HIV positive and not have AIDS for sure, but you can't have AIDS without having HIV. We're going to keep that under control with antiretrovirals. Yeah, avoid sexual contact, especially use protection if you are having sexual contact, barrier methods. And we have medications that are antiretrovirals that can bring people down to 0 viral load, but you should still be using protection because they tested 0 virus, but that's undetectable. But that doesn't mean they don't have the virus still, which in that blood sample, they didn't have it. So we're working on vaccines and other drugs to treat it, but right now we mostly target that reverse transcriptase and other proteins that are involved in the viral life cycle that insert the DNA into the genome or prevent it from making new viruses. And that's how that'll work. All right, so that is it. I'm going to leave it here. Go over the class questions in your own time. If you have any questions about those, please let me know. We're going to go up to the lab and do our last lab. Great discussion today, everybody.
I'm going to go back in.